Hi there. Today I'm going to show you how to build co-expression network from gene expression data. This is going to be an R tutorial, and in order to follow it, you will need to have R and Rstudio installed. The main program that we are going to use, or package, to analyze the data is the R package igraph. But in order to interactively visualize the network that we're going to get from igraph, we're going to also need Cytoscape. So you will need to have all these three programs downloaded and installed according with the requirements of your operating system. The tutorial is very simple, so you should be a beginner of using R and Rstudio, so you just need to copy and paste the commands that are in the tutorial. The entire tutorial has been deposited in a GitHub repository, which I'm going to link in the description below. Also, I have provided an HTML file where you can not only see the commands for running the tutorial, but also the expected output that you should be getting from running the commands. Okay, so what we are going to start with, we are going to start with gene expression matrix. In our columns are going to be the genes, and in our rows are going to be the samples. Once we have this gene expression matrix, so it has to be with normalized gene counts, we are going to have all the components that are going to be interacting, and these are going to be our genes. And at the same time, we're going to be able to obtain the similarities between the expression levels of the genes that are present in this expression matrix. And this is going to represent the interactions between our genes. So from this, we're going to end up having this. And what kind of specific questions building of interaction networks starting from gene expression data can address? So we can identify new biological mechanisms that, for example, we cannot identify if we look at individual components alone of this network, such as new pathways or new multi-protein complexes. Additionally, we can identify master regulators of a process, for example, a transcription factor that is overrepresented amongst all molecules that are found in the interaction network or individual subregions. And not only transcription factors, but also genes that are important for the onset or development of a disease. And one can deem this as disease targets and start to develop drugs against these targets that have been identified in this way. Another fascinating property of interaction networks is that it can help us identify molecular functions of genes or molecules that we have absolutely no idea what they are doing. For example, simply by finding very strong interaction patterns between a molecule of a known function with many other molecules that we already know the function of, we can infer that likely the function of the molecule that we have no idea what it is doing is the same as the molecule that it is tending to form very strong and a lot interaction with in the interaction network. So this is called a guilt by association principle, and is a very powerful concept of interaction networks. Okay, but let's now explain some basic terms of network theory. It's important to mention here that networks and graphs are interchangeable terms, depending on who is talking about networks or graphs. So mathematicians are calling them graphs, and sociologists are calling them networks. But if you hear me talking about network or graph for the rest of the presentation, it means exactly the same thing. So because we're talking about co-expression networks, apparently the entities that are forming interactions in this context are our genes. And the entities that comprise a network are actually called nodes. So we have gene 1, gene 2, gene 3, etc. So you can see the genes here as well. So another important entity of the networks is an edge. And the edge is a relationship between nodes. So this is what it represents. And here, these are the lines that you see. These are the co-expression patterns or presence of similarity in the expression between two genes that are linked with this kind of line. Also, it's important to mention here that edges can also have a property of their own, that is, it is called weight, and it represents the intensity of relationship that they are establishing between two nodes. Sometimes it is represented by the difference in the thickness of the line that is connecting the two nodes, for example, like here. But in other times, this can also be represented by different colors or different ranges of colors, so and so forth. And another important thing, nodes are not only called nodes, they are also called vertices. And you can hear that edges are sometimes referred to edges, but other times referred to links. And another important thing about the basic concepts of graph theory, so if the weight of the edges are specified, or the intensity of those edges, the network is called weighted. And if the weight of the edges is not specified, or just their presence or absence, then the networks are called unweighted. So we can build a network via integrating its nodes and edges. And then by analyzing the properties of the nodes and edges in the context of network, we can identify how important particular nodes or edges are for the maintenance of the structure of the network. For example, one property of the network that is very famous, especially property of the nodes, is that they can have varying number of interactions with other nodes. And if a node has many interactions with other nodes in the network, it is also called hub node, or in our case, hub genes. And I'm sure that you have heard this term hub genes before. But the whole notion of counting the number of nodes that one particular node interacts with is called degree, or degree centrality. So nodes with very high degrees are important for the maintenance of the structure of the network, and if you remove them, it means that the network is going to disintegrate. So another property of the nodes is their betweenness measure. So I'm not going to go much into details about that, because I want to make the introduction relatively short, but the higher the betweenness of a node, or the number of shortest paths that go through the node and connect different parts of the network, the more important is this node for connecting different parts of the network. For example, this node 1 here, if we remove it, it will lead to disconnection of the entire network because it's quite central and it has very high betweenness. Another node that has very high betweenness is this number 2, but it is actually connecting two local communities rather. Okay, so not only the nodes or the edges can have properties, the entire network as a whole can also have properties. For example, we can estimate the mean degrees for all nodes in a network, and that's going to be a value that we can compare this network to completely other network. Another property of the network that we can use to compare one network to another is average path length, which is going to be only one number. Another one is node degree distribution, which is a graph, and I'm going to talk about it in the R tutorial itself. Another number that we can compare is clustering coefficient of one network to a different one. Okay, a very important property of a network that all biologists, I would say, are obsessed with is the formation of local communities in a network. And what local community is basically an area of network where the nodes tend to form interactions with themselves much more than with the rest of the networks. For example, in the example network that I'm showing you here, we have 1, 2, 3, 4, 5 local communities. They're also called modules of co-expressed genes in the context that we have. Okay, and normally people would look for biological processes that are enriched in these individual local communities with co-expressed genes. But how are these analyses different than actually looking for overrepresentation of biological processes in differentially expressed genes, for example? Okay, let's imagine we have this scenario. So we have this is gene expression network. These are our samples, these are our genes. In order to deem a gene differentially expressed, it just needs to have a pattern of expression that is shared between minimum number of samples. For example, if you compare disease to healthy, it has to be shared between at least 3 disease samples relative to a few of the healthy controls, and it means that the gene is going to be deemed as differentially expressed. For example, another gene can be deemed differentially expressed having the same direction of differential expression, but having completely 3 different samples that are having this pattern shared between them. And it means that in order to deem a gene differential expression, we do not need to observe the pattern of differential expression across all samples for a particular condition. If we would like to take the same data and identify co-expressed genes in this data, we have several options. We can take the healthy and the disease all together and look for co-expressed genes, or we can separate the disease samples and the healthy samples and look for co-expressed genes in either of those. What co-expressed genes would mean if we decide to do the analysis this way? So we would need to have, for example, A and B gene. We are going to look whether they're co-expressed or not. Their pattern of expression should be shared, or variability of expression should be shared between all samples of a particular condition or the entire data frame, depending on what we decided to work with.